So the first group of biomolecules we're going to focus is the proteins. Now, we talk about proteins first. It's because proteins are so important. They're one of the most abundant organic molecules in living things, living organisms, or catalysts, right, to speed up chemical reactions. So normally, those chemical reactions will proceed very, very slowly under normal conditions. But when you provide enzymes, they will go really, really fast. Okay, so our body is undergoing, hundreds, thousands, millions of chemical reactions. So without enzymes, those chemical reactions probably cannot be achieved in a very short period of time. So enzymes are very important. When you eat food, you need those enzymes to kind of speed up the breakdown of the biomolecules in your food. Okay, so that's one example. We also have proteins that can transport different substances. Okay, a good example is hemoglobin in your red blood cells. Okay, I don't know if you know the function for red blood cells. They transport gases, right? Very important gases. For example, oxygen and carbon dioxide, right? Without oxygen, you can't live, right? Your cells will die. You also need to transport the carbon dioxide away from your cells back to your lungs so that you can exhale and get rid of the carbon dioxide. So the transport of oxygen and carbon dioxide is done by hemoglobin in your red blood cells. All right, another good example, some of the hormones are proteins, and you know how important hormones are, right? So for example, insulin, it helps our cells take in glucose, right? And use glucose as an energy source. So if you don't have enough insulin, or your body doesn't, makes very little insulin, you're gonna have diabetes, right? Your blood sugar level is too high, and that's gonna damage some of the tissues. Next one, defense. Another very good example. So when you get vaccinated, your body will produce antibodies, right? So next time if you're exposed to the same pathogen, those antibodies will be stimulated, very, very fast, and they will protect you from those pathogens. Okay, so those antibodies are proteins. All right, contractile proteins. You move all the time, right? And how do you generate movement? Through your bones and muscles. Now, you have to contract your muscles to move your bones, and it's achieved by the protein fibers, including actin and myosin in your muscle cells that will move, right? Interact with each other, move, and that's what leads to muscle contraction. Okay, so you can see proteins are so important. Now, proteins are polymers, and they're made of monomers, right? They're subunits, and the subunit is amino acid. So you absolutely have to remember that proteins are polymers made up of monomers, which are amino acids. So here's a picture showing the structure of a typical amino acid, and this is important. I will probably have the structure on the test to kind of have you identify the different groups and some characteristics of amino acids. All right, so where do we start? Now we're going to start from this central carbon. Okay, so this is the carbon that's right in the center. And then you're going to add 4 different things to this carbon. Now, carbon can form up to 4 covalent bonds, or 4 covalent bonds. And then we're just going to add 4 things to it. Okay, and that's going to form amino acid. The first group you're going to add to that central carbon is amino group. You probably still remember it from last week's lecture, right? Nitrogen and 2 hydrogen. So that's amino group, and that's part of the name. And the other big group is going to be the carboxyl group. So the COOH. And if you remember, this hydrogen can dissociate, right? It can leave this oxygen, and when it gets into the water, it becomes hydrogen ion. So that's going to make the solution acidic. Okay? So this is the carboxyl group, and this is the acid part in the name. And then the third thing you're going to add is a very simple one, just a hydrogen atom, and that's it. And then the last group is going to be R, so this is the side chain. Now R can be different things, and that determines how many amino acids that we have. So before I show you the different kinds of amino acids, we are going to identify a couple things. Okay, first of all, the end with the amino group is going to be called the N terminal. Okay, N-terminal. Let me go back to here. So that's where this term comes from. And then this end with the carboxyl group, that's going to be the C-terminal. Okay, C-terminal. Okay, so this is how you can identify, kind of different ends of proteins. And then if you see essential amino acids, those are some. Okay, so we have 20 amino acids, 20 different kinds of amino acids in our body, and our body cannot produce. Okay, we don't have the ability to produce about half of them. So those amino acids are considered essential amino acids, okay? Meaning your body cannot produce it, so you have to get them from diet. Okay, now we know amino acids are monomers, so you need to link them together to form the polymer, right? Okay, so this is how it works. Remember that dehydration synthesis, right? So this is amino acid, this is a second amino acid, and then when they want to be connected, they each lose either a hydrogen or a hydroxyl group. So that forms a water. Water is gone, and then these two atoms, the carbon and nitrogen, will be connected by a covalent bond. Okay, so this is how you can link all the amino acids to form a longer chain, which is polypeptide. So that longer chain is going to be polypeptide. Okay, and then this chemical bond between the carbon and nitrogen is called a peptide bond. Peptide bond. So remember that the bond that keeps the amino acids links is called a peptide bond. All right. Okay. Now let's look at different amino acids. Here I put a note here. Let's see what we're going to do first. Let's look at each group. So here we have a nonpolar amino acids. So we're going to talk about why these amino acids are nonpolar. This is kind of more or less like a review of the functional groups that you just learned last week. Okay, let's look at these amino acids. So the R group, the side chain, is highlighted in blue. So you can see all the amino acids have the same 3 of the 4 groups, right? The amino group, hydroxyl group, and then this hydrogen. So the only thing that differ from each other is this side group, the R group. Okay, so the smallest, the simplest amino acid is going to be glycine, because the R group is really just a nitrogen atom, and that's it. Okay, so it's very simple. And then you're going to kind of build up. So alanine is going to have a methyl group, right? CH3, that's the methyl group. Okay, and then valine is, you can see, you're going to have 3 carbons, right, instead of 1, and then more hydrogen atoms. Okay, and then when you get to, let's see, here, this is a pretty long chain, right? So you have 1, 2, 3, 4, 5, 6 atoms in that backbone, and then over here you have some big, carbon rings, which, these amino acids are even bigger. So let's look at why some of the amino acids are nonpolar or some of them are charged. Okay, so you only need to focus on the R group. So when you see hydrocarbons, so basically it means you have mostly just carbon and hydrogen. Carbon and hydrogen. Okay? So it can be as small as a methyl group. It can be very, very long, like these are pretty long. Okay, it's going to make the molecules very nonpolar. Okay, and then that's almost equivalent to hydrophobic, right? So these functional groups are very hydrophobic, meaning they fear water. They do not like water. They don't want to mix with water. So these amino acids do not dissolve very easily in water, so they're nonpolar. And then over here, this is also a nonpolar amino acid group. So what happens here, instead of more linear chains, you have these carbon rings, right? Could be 5, could be 4, could be 6, like this benzene ring. So these structures, these ring structures, will also make the molecules nonpolar, because these functional groups are very, very nonpolar. They're very hydrophobic. So these amino acids are considered nonpolar. Okay, all right. When you look at the polar amino acids, usually they have extra charge, okay, on the structure. So the amino group carries one positive charge, and then the hydroxyl group carries a one negative charge, so they kind of cancel out, right? So you just need to look at the R group. When you have a hydroxyl group, remember hydroxyl group is very polar because the oxygen is very strong in kind of moving the electrons towards themselves, right? So you have negative charge on the oxygen end and then positive charge on the hydrogen end. So when you have unequal distribution of electrons, which results in a positive end or slightly positive end and slightly negative end, then it's very polar. Because of the polar ends can interact with the water, so they're polar, they like to dissolve in water. So every time you see hydroxyl group, that means the molecule is probably kind of polar. Okay, and the same thing for sulfur. So the sulfur is kind of like oxygen, right? It's kind of stronger, compared to hydrogen in moving the electrons, closer to themselves. So that will create a slightly positive and slightly negative end. And when you look at these two groups, so now they're positively charged or negative charged, so they are definitely polar. Right? So now the question is what kind of charge they carry, right? So these are positive, you can see the positive end, and then these are negative. So basically they have an extra carboxyl group to carry that extra negative charge. Okay? So these are definitely polar. So on the test, I'll give you a few examples of amino acids, and then I will ask you for their characteristics, right? So if I give you something like this, I'm going to ask you, is this a polar or nonpolar amino acid? So your answer will be nonpolar, okay? Or if I give you an example like this, and I'll ask you whether it's polar or nonpolar, and the answer is polar, right? Because there's an extra positive charge here. So since it's charged, it's definitely polar. Okay, and I may ask you more if it's a polar, is it charged or is it uncharged? You're gonna say it's positively charged because of that positive charge, okay, on the nitrogen part. Okay, so these are different characteristics of amino acids. Again, everything mostly depends on that R side chain or in the structure. Okay, so now we're going to look at protein structure, and then it's going to be on 4 levels. Okay, so remember proteins have a really diverse range of functions, and that's because they have very diverse structures. So we're going to look at 4 levels. So first level is going to be the primary structure. So the primary structure really just refers to the unique sequence of amino acids. This is bovine insulin, so it's a protein insulin. It's kind of hard to remember that insulin is protein. You tend to think that it's probably some kind of lipid, but it's actually protein. Okay, so this insulin molecule has 2 chains. Okay? Now, when you look at each chain, see each bead or circle indicates amino acid, and usually we just use like a 3-letter system to indicate which amino acid. So this is going to be the sequence, right? Glycine, isoleucine, valine, glutamate, blah blah blah. Okay. And then the B chain has a different sequence. Okay. Now, just remember, if you have a different sequence, right, different amino acids, their R groups are going to be different. So those chemical reactions between those R groups are going to determine, the higher tier structures. It's ultimately determined by your gene sequence, right? Okay, so if you have a mutation or some kind of damage to your DNA, it's going to change the primary structure or the amino acid of that particular protein. Okay? So a very good example is the sickle cell anemia. So you guys probably have heard about this disease. Unfortunately, it's very prevalent in African Americans because this mutation originated from Africa. Okay? Patients with sickle cell anemia a lot of times can experience very extreme pains, very excruciating pains. So I've heard some really terrible things about this disease. Now all that is caused by change in one amino acid. When you have a mutation, it's gonna make valine, this amino acid, instead of a glutamic acid. Okay? Now, because of this one amino acid change, the structure of the entire protein changes. So instead of a more globular structure, the abnormal proteins tend to kind of form clumps, aggregate. So they're going to form a more linear structure. So instead of normal red blood cells, as you can see over here, okay, you are going to have a sickle-shaped red blood cell. And that's just because the hemoglobin has a different structure. Okay? And these sickle cells do not perform the same as normal red blood cells. So they're very rigid, so it's very difficult for these cells to pass through the tiny capillary blood vessels, and a lot of times they tend to break, right? And then you have anemia. Okay. And again, remember, this is a very good example of how important the primary structure is. Okay. Now, secondary structure. So imagine you have a long chain of amino acid, so that's called a polypeptide. Now, in certain spots, you may have local folding. Okay? So see, this is a very long chain of amino acid, and then at this spot, you may have amino acids forming an alpha helix, a helical structure. Okay? So think about those kind of helical shaped stairs, like in a very fancy house, like in a mansion. Okay, so that looks like that. A different secondary structure is the beta pleated sheet. Okay, so at this spot, you may have, amino acids forming hydrogen bonds in between them. So there are two lines, right? And then you have a bond in between. So that forms a sheet-like structure, which we'll call beta pleated sheet. So the local folding of the polypeptide chain, the amino acid chain, is it gives rise to the secondary structure. Tertiary structure. So now, instead of a near polypeptide chain, like a linear amino acid chain, now the chains start to form a three-dimensional structure. Okay, so again, instead of linear structure, the chain can fold, right? Certain spots may pop out a little bit, right? Kind of fold. So this forms a three-dimensional structure. So how can you get a linear structure to form this 3D structure? Now that's all because of the chemical reactions between the amino acids, specifically between their functional groups, the side chain R groups. So I'll give you an example. Over here you have ionic bond, right? So if you have this R group and then a different R group over here, so the amino group carries a positive charge, right? And then the hydroxyl group carries a negative charge. So if you have amino acids that one of them is positively charged and the other one is negatively charged, then their R groups are going to be attracted to each other, right? Because opposite, charges they attract. So it's going to kind of fold the chain this way, right? Because you're going to have a chemical bond link over here. Okay. Another incident is going to be hydrogen bonding. So this is the hydrogen bonding. So again, you can see that it may make this part kind of fold back a little bit. Okay. This one is pretty important, the hydrophobic interaction. So this is going to be between hydrophobic or nonpolar functional groups. Okay, so nonpolar, you guys remember, they don't like water, right? So these the amino acids with these functional groups, they tend to stay inside in the interior of the polypeptide chain. They don't like to be out because we have more or less watery environment, right, inside our body. It's mostly water, so they don't like water, so they want to stay inside. And then amino acids that are polar, they tend to stay outside on the exterior, so they can interact with the water. They like water. So this, again, is going to contribute to that 3D structure. Okay? And the last one is going to be disulfide linkage. So some amino acids have sulfur in their R group, and then the two sulfur atoms can form this disulfide linkage. Okay, so again, this can cause the chain to kind of fold around, right, and then form a 3D structure. The last level is going to be the quaternary structure. Okay? Now, some proteins are formed from several polypeptides known as subunits. Now, I'll highlight it here. So the subunits here should not be confused with the subunits when we talk about polymers and monomers, right? So in that slide, we refer monomers as subunits of polymers. But this is different. This refers to each polypeptide chain. Okay, so what is a quaternary structure? We have to go down to this slide. Okay, so you have the three previous levels we discussed, and then the last level is the quaternary. Okay, so you can see there are two chains, right? Because they're in different colors. So this is one polypeptide chain, and this is the second one. So what's the difference between polypeptide and protein? Okay, so some proteins may have only one polypeptide chain, then they're the same. But most of proteins, they have multiple polypeptide chains, right? Like in this example, this protein has two. And then when we look at insulin, right, that's also two. But when you look at hemoglobin, I don't have a good picture here, there are 4 polypeptide chains. So for those proteins, the 2 polypeptide chains, or subunits, will interact with each other, and then they form this entire protein structure. Okay, so that's the difference between polypeptide chain and the protein, and then this also is about the quaternary protein structure. Okay, polypeptide chains interact with each other. Okay. Proteins can be damaged. Okay, once the structure is changed, altered, then their functions will be altered. Okay, so what is denaturation? Denaturation. If a protein is subject to changes in a lot of things, temperature, pH, or exposure to some other chemicals, then the protein structure may change. So they probably lose their shape and lose their functions. Okay, so that process is called denaturation, right? They're losing their nature, their structure, their function. So denaturation may be reversible or irreversible. Can't talk today. If it's reversible, then that means the protein can go back to the normal shape, so it can still perform the functions. But if it's irreversible, then the protein pretty much just loses its function. Okay, I'm going to show you some examples of where denaturation takes place and it's irreversible. All right, so you're familiar when you cook meat or shrimp or egg, the texture changes, right? So this is a thermal denaturation because you're using heat to denature the protein. Okay? So this is irreversible, right? You can't make these go back to what they looked like originally. You can also use mechanical force to denature protein. This is not a very good picture, but think about the time when you use heavy cream to make whipped cream, right? If you make a pecan pie, my husband makes the best pecan pie for Thanksgiving. Can use a lot of force, right? Just keep stirring that heavy cream, and eventually it will kind of more or less solidify, right? And it forms cream, whipped cream. This you might not be familiar. This is how we make tofu, right? So you have a soy milk liquid, and you add some salts, and that's going to change the protein. So it solidifies and it forms tofu, like the tofu cubes, right? Yeah. Okay. So this is very interesting about resistance of proteins to temperature. Now, the previous examples, all the proteins are destroyed, right, under high temperature. So this is a very interesting. So if your water is not clean, you can boil the water, right, to kill the microorganisms to make it safer. But certain bacteria, they're in the archaea group. If you remember, that's one of the domains, right, among all the 3 domains. So bacteria, archaea, and then eukarya. So bacteria in this domain, they can be very resistant to high temperature. Okay, so there are some bacteria, they are considered hyperthermophilic. Hyper, very high, thermal temperature, philic means love, right? So they love hot temperature. So you can find those bacteria in very extreme environments like this hot spring. Okay, so we just look at we just looked at a lot of examples where heat can destroy protein. So what happens to these bacteria? How come they're not damaged, right? So their proteins can still function. That's why they're still alive and they live happily in those very hot environments. Okay, so this is how it works. If you have some mechanisms to make your protein stable, meaning you can maintain that particular shape of the protein, then your protein can be resistant to environmental factors. So these bacteria have proteins that can be very stable under high temperature. Okay. Now, what makes the protein structure? Remember the different levels, but ultimately it depends on the chemical reactions between the R side chains, right, of different amino acids. So for these bacteria, they have a few things that kind of help their proteins maintain the shape, even under very extreme temperatures. So first force chemical reaction is this hydrophobic interaction. So you have groups or amino acids with hydrophobic R chains. So they like to stay deep within the structure. They don't like to be out. They like to be inside. And these amino acids or this kind of chemical reaction helps maintain a normal protein structure. Okay? And also, the proteins of these bacteria tend to have a lot more charged residues and salt bridges on the surface. Okay? So all those things, if you remember, charge, right? Opposite charges attract. So that helps keep that part folded, right? And then same thing for the salt bridges. So because there are a lot more chemical reactions, they are going to help maintain that regular protein structure. So that will make sure that these proteins still perform the normal functions. Okay, so this is why these bacteria can live in very extreme environments. Okay, so we talk about the different levels of structures, and we know that correct folding is very important for proteins. So a lot of people don't realize that, but proteins need assistance in the folding process. So they have some helpers known as chaperones. So the chaperones are proteins themselves, but they can help other proteins, the target proteins, to fold correctly. All right, so without chaperones, you don't get correctly folded proteins, and you're not going to get that function. Okay. So remember, if I ask you what molecules can help proteins in the folding process, make sure they are folded correctly, your answer will be the chaperones.